Welcome everyone, thank you very much for joining this presentation. I will give you an update on the work that we've done on the organoid technology this last 2 years. Starting with a short introduction. That introduction I usually start with this slide that indicates where we think the organoid technology has its impact, where we think the organoid technology contributes to drug development. And it is basically this problem that, of course, everyone in drug development knows, that when we do our preclinical work, the models we use, animal models or cell lines, are useful to show biological function. But in the end, when we look at clinical trials or the drug use in the market, we find that as is understandable, that these models are not patients and therefore are not predicting very well what the patient response will be like when we actually use it on large groups of people. And this leads to this relatively inefficient drug development strategies that we are used to, expensive drugs. Whereas we think that if you improve the preclinical models, if we can improve the patient representation in the preclinical drug development, we would significantly increase the efficiency of drug development and thereby decreasing costs. Organoids fit exactly in this, because and also why we call it patient in the lab. Because what we do with organoids is basically we can take samples from patients and directly put it into the lab to do our research on. And that all started with the discovery of the adult stem cell more than 10 years ago by now in the lab of Hans Clevers. This discovery of the adult stem cell initially on the intestine and then other organs allowed us to isolate this cell and to try to understand it better and to see if we could find means to take this particular cell and expand it in vitro. And this is indicated on this slide. You see in the left top corner that we can start with single cells, or as we now do in the lab with tissue fragments, but initially with single cells, to make the point that indeed when we take out this single adult stem cell and we provide it with the right information, with the right growth factors, we can expand this cell unlimited, and very importantly, with keeping its genetic stability. So this is the problem with cell lines, of course, that when we grow it in the lab, we get the adaptation to the culture conditions, and thereby this genetic and also epigenetic modifications basically mean that the cell that we're growing in the lab is no longer the patient cell, and thereby, although it is useful to identify biological function, it doesn't necessarily mean that the responses are like our patient-like. So the interesting thing in this method is that indeed, when it was Sato in the lab, when he was able to take this stem cell and to expand it, we not only got a lot of stem cells, but we in fact got a structure that is, you see it here, it's a little bit of a fumbled up intestine. But what it is, if we look at it microscopically, is a real epithelial barrier of an intestine, where the luminal side of the intestine is on the inside of the organoid, and the outside of the organoid is the basal membrane of the intestine. And that also goes for other organs. If we use lung or breast or other tissues, that works in exactly the same way, that the luminal sides are inside and the other side is on the outside of the organoid. And the cells in the organoid have stem cells, differentiated cells, function and communicate together as if there are a normal epithelium. So with this, and in combination with what I already said, the genetic stability, we now have a culture system that has a functional resemblance to normal tissue, while maintaining this genetic stability on the long term. Because another very important quality is that with this method, we can expand these cells virtually unlimited, depends a little bit on the organ, but without having the intestine here. So we can generate biobanks, we can generate reservoirs of these cells that can last us, as cell lines do, for an indefinite amount of time. And thereby we can capture patients long term, we can capture the heterogeneity of different patients, and we can study these stably over an extended period of time. So that is by now 10 years ago. So this is, as said, we started with the intestine, and having these intestinal stem cells, we then started working on other organs. The epithelial systems, epithelial stem cells, turned out to be fairly similar between cell types, between organs. So this means that with these intestinal system, we could later on develop also lung, liver, pancreas, the organs that you see represented in this video. Importantly, we can take, when we talk about cancer, we can take healthy tissue, we can take diseased tissue, as you see here, cancer and healthy, which basically means that we can generate an internal control of patients, and we can expand these cells, cancer stem cells now in this case, stem cells and the normal healthy cells indefinitely. We then characterize these cells, genetic characterization, of course, genetic being the basis of most of the work that we're doing in the lab, is added onto the system. But, and that is the important organoid attribute, we now have a functional, a living cell, so we can do functional experiments on all these patients. So we can combine basically what we know from before, genetics, clinical data, with functional data on each organ and see the patient-specific drug responses. So that was the key to the organoid technology and the basis of it of a couple of years ago. Of course, this claim that the organoids are indeed a model that is closer to patients and to be supported, because we understand all that a key aspect of clinical work is that we want to have a model system that is patient-like. However, we need to prove that. And an important part of organoids is that indeed now you can, whereas in the past genetics or other data are indirectly related to potential patient outcomes. What we can do with an organoid, because we isolate tissue fragments from individual patients while they're in the treatment process, while they're in the hospital, we can take these samples. You can see that here for an example of breast cancer. So you can take an organoid sample, a biopsy. We can generate an organoid from this biopsy. We characterize that in the lab. There's genome sequencing, drug screening, etc. Basically, any sort of technology that we generally use in the lab. And then we can take those organoids, and you see that here in the left bottom, and we can test different patients. For instance, a patient here represented in green and one represented in red. And we test the drugs that are also used on these same patients in the clinic. And what we find is that indeed, and that the importance to the previous claim that is it mimicking patient responses, what we see is that there is in green a patient samples, organoid samples that are responding very well to this hormone treatment, whereas we see in red patients that are not responding. And now when we go back to the clinic, what we see is that also when we look at the same patient from which we took this biopsy, from which we generate these organoids, we see that that same therapy that we used in the lab, the responses are the same. So we see in green a patient that indeed clinically responded, whereas in red we have a patient that did not respond. I think this is the key to organoid experiments, the way that HUB is generating organoids, but also the way other labs, academic and commercial labs are generating the 21st century models, let's say. That indeed, this is for the first time this type of work is possible. So now we can take patient-specific samples, use them in the lab, and have a direct correlation between what is happening in the lab and what is happening in the clinic. And this is I think the basics of how we can use these new models in the future for drug development, knowing that we should actually, when doing our preclinical work, we should try to make this direct connection and use it as if we are looking at a sample in the clinic. So I have this is summarized here. So they're basically meaning that traditionally we've had clinical data and molecular data here on the left. With organoid data, we basically add a functional test to each laboratory experiment and to each patient that we can use to combine the clinical side of our work to the preclinical side of our work and use it either in identifying biomarkers, patient stratification, drug development, but even in potential diagnosis support, so predictive diagnostic testing to determine what type of treatment patients might need. So that's what I wanted to show you as an example, as an introduction, and this is the basis. I think this is also what has to come back in this model development in our field, that we constantly consider when we are generating nice biological systems. Do they in fact represent what is the patient, and that can now be validated. So it's important to do that. In this graph, you see what that means. So now we have on the one hand shown that when we have an organoid, we see that these responses in a large part represent what happens in the patient. So when I have a cohort of patients in the lab, organoids, and I use in this case, for example, 4 different treatments that are used in the treatment of colon cancer, I can have an assumption of how these patients would respond in the clinic. So you see indeed that when you take here in the different colors, 5FU for instance, that we see this range of drug responses as expected in the clinic, also back in the organoids. And that basically goes for the 4 treatments that are represented here. The different patients show a different response, and that you see this heterogeneity in responses among patients as we expect in the clinic. So that as an introduction. So what I wanted to show you is that with this we have developed different models, and also what is an important aspect at the moment that, as already discussed, on the one hand we have this clinical validation. On the other hand, I showed you this initial organoid, this sort of 3D cystic structure that represents the epithelium. That is the basics. That's, let's say, the first type of organoid that we can use for especially viability screening, drug screening, as I showed you. However, for certain other diseases or interaction with the immune system, we need to develop new organoid models that have other ways of representing the epithelial barrier, and those are the examples that I want to show you today. So this is an example of lung, which is where the initial sort of 3D organoid is capable of already showing you quite a lot of a phenotype. In this case, we are representing the cystic fibrosis phenotype, because what you can see here is that in the normal the normal organoid here, this is this is not a video, but if you would look at it in the video, you see the cilia present, but also beating. But what you see on the bottom is that we have a healthy organoid and an organoid from a cystic fibrosis patient. And as expected and as seen in a cystic fibrosis patient, is a very thick mucus layer present in the cystic fibrosis airway organoid, whereas this is absent in a normal organoid. So you see that these organoids represent, on the one hand, the genetics and the genetic stability, but also the phenotypic characteristics that we see in several diseases can be represented, as here with the secretion of this very thick mucus that we know from cystic fibrosis. Here's another example that in part by the labs at Erasmus University and Cleves using the organoids in viral infection, of course very relevant at the moment. On the right we see an older example from Huch, where he used the lung organoids to study RSV infections. This is an important one because that indicates that the gene expression again in the organoids is as it is in vivo, because cell systems are often difficult to recapitulate viral infection because the gene expression, the protein expression on the membrane is not sufficient to support viral expansion. Whereas here you can see that indeed we can infect a lung organoid with RSV, and we have a stable long-term infection of these replicating virus in these organoids. In fact, when we use a specific antibody that blocks the uptake of the virus, we see indeed on the right here that the GFP in the right top is absorbed in the normal organoids, but when we add the antibody, the RSV, GFP labeled RSV is not taken up by the organoids. And on the left, of course, the very relevant example at the moment is that indeed you can use, in this case, intestinal organoids to study, for instance, corona infections as well. And as was shown in a study earlier last year by the Cleves and the group in Rotterdam. So I think the importance of this is, other than that we can use it for viral infections, is that it is an indication again that organoids have gene expression patterns that are very similar or are similar to normal healthy tissue, and therefore can be used for studying a range of biological functions, for instance, viral infections. Another important one is that we often use as an example in toxicology, which is another preclinical field in which the healthy systems healthy cells are of course very important, but also very important is that there is natural mature gene expression in the systems, which is sometimes absent. For instance, when we look at embryonal derived stem cells or iPS derived cells, in this case, because we are working with an adult stem cell, we can generate these mature cells, both stem cells but also differentiated cells, as I am going to show you. Another important point is that the organoids, actually the first organoid was developed from a mouse, and before we went to develop the human system, In toxicology, of course, a lot of animal species are still being used. What we've seen in our experiments is that the organoids from these animals, for instance, mini pig-derived organoids or dog-derived organoids, show the same phenotypes as we see in the animals themselves, similar to what we see the clinical representation of human organoids, of course. So that means that for these particular toxicology studies, we can use animal-derived organoids, human-derived organoids, put them together, and have a more complete picture based also on historic data that is often animal derived, to basically assess the safety of specific compounds. And on the right, an important point, because we have much more freedom to choose which type of organoid we need, as opposed to cell lines that are just sometimes randomly derived and when it works. We can develop organoids from different parts of intestine or colon or other organs, because we know that gene expression and therefore safety with toxicology responses are different in different regions of organs. So I'll just again give you some examples. So in this case, we studied two molecules, gefitinib and this molecule here on the right with the difficult abbreviation. So in this case, what we use is different animals, dog and the rat, and then comparing it to human. And the key point here is that what we see, and known for this particular molecule, is that we see a viability response. So this is the viability testing that is representative for what we have seen in people and in animals as well. So where here we have a compound that seems relatively is quite toxic in the dog and the rat, whereas we see that in humans it's actually not very toxic at all. So when we test these animal models in the lab in a preclinical phase, we actually are maybe giving the wrong message, because gefitinib is not as toxic in the human samples as it is in the animal samples. Whereas when we look at this particular molecule, we see the reverse. And we see that although it is quite toxic in the rat, it is much less toxic in the human. I'm saying this the wrong way around here. So indeed, we see here that the human sample is quite toxic and the animal samples were not. Whereas here we see that the toxicity is seen in the rat, whereas we see that it's largely absent in the human sample. So basically, what it tells us is that we can use these animal organoids, but also therefore the human organoids, to get a better picture. And a picture that is often skewed when we use animal models because of the difference in the species. Another important point is that, as I alluded to earlier, is that this is done when we use the 3D type organoids. But for many type of assays, it's actually easier to have it in a normal, as in the intestine itself, access to the different sides of the membrane. So we developed also the same organoid structure in a 2D system, so transwell-like system. Something I didn't talk about before, but based the media conditions based on the growth factors that we are adding, you can steer the differentiation of the organoid towards stem cells or towards the different differentiated cells, as you can see here. That changing from this to this, for instance, we see the expression of mucins, which is absent in this type of media where we focus more on stem cells. So basically, in the colon and in the intestine, by using different media and different setups of the systems that we use, we can gear the model towards whatever experimental needs or biological means we need to study. And you see an example here then for what that means. So we use the transwell systems, as in the previous slide, for doing a tear assay to basically measuring electric conductance over the membrane, which means that if the membrane is disturbed, for instance, that happens in IBD, inflammatory bowel disease, that in that case the resistance of the membrane obviously decreases. And you can see that here we mimic the inflammation phenotype of the disease by adding cytokines. And so in black we see that we have a membrane that's intact, that has full barrier function. But when we increase the cytokines that we can see in an inflamed intestine, we see that indeed this causes the barrier function of the membrane to be lost. Here by the decreasing resistance increasing over this membrane. And then interestingly on the right, if we use a particular compound that inhibits this phenotype, as we know from the clinic, we can restore the barrier. So basically here we introduce in the graph to the bottom the high concentration that we found on the left of the cytokines, and when we add tofacitinib, then we can restore the barrier function of this intestinal organoids. So basically, with this, we mimic the inflammation, and we can study compounds that actually restore barrier function in the intestine. This is, as I said, something that we use for IBD. This is an example of that. So indeed, when we want to study inflammatory bowel disease, we can isolate organoids from, and you see in the middle panel, from tissue itself, you see the inflamed colon tissue, and then next to it a normal tissue, and generate organoids from both. We see gene expression to be different indeed in these two regions. Of course, as we know, some of this is lost in time, because when the inflammation goes in a culture system, also a part of that inflammatory signature goes. However, other parts of it are more stable, and you will see long-term in the organoids. So with this, by taking different regions, as said in the beginning, and by taking different regions of the intestinal colon and other organs, of course, we can study the specifics of that region. And also, in the case of this inflammatory disease, we can take inflamed region organoids and healthy, relatively at least, and uninflamed sections of the intestine, and make organoids of both to do comparisons. So with it and another important organ, and when we talk about the maturation of cells for toxicology studies, is the liver, of course. And that was an important one for us also to establish whether the organoids can be mature enough, with C and IPS or ES-derived hepatocytes in vitro, is that they don't reach the metabolic function, they don't reach cytokine expression, as we see from normal hepatocytes. So what we did with the organoids, and you can see that here on the left panel, we create this expansion state in which we have mostly stem cells that are growing. And then from that, we go to a second system where we differentiate them into hepatocytes. And you can see that from 7 to 15 days, you get an increasing expression of, in this example, CYP3A4. Showing the maturation of the hepatocytes. And what is important on the right is that we see again the expression of, and this is actually enzyme function, this is an enzymatic assay of cytochrome 3A4, but also 2C9. So we have the different cytochromes expressed. And in the bottom, very importantly, when we compare that to donor hepatocytes, so hepatocytes that were derived directly from the liver, we see that, although with a short delay, we see to the right of that graph that when we look at, in this case, midazolam metabolism, basically the ultimate readout of the maturation and function of these hepatocytes, we see that we reach a level that is very much the same between an organoid-derived hepatocyte and a normal donor-derived hepatocytes. So very different from IPS and ES, which would be much close to the bottom when we look at at this particular metabolism. So indeed, we have hepatocytes that are seemingly mature, and then what we can see if we test again, this is a viability assay. As an example, when we test different compounds that we know from the clinic are toxic. So here we have in the blue and red 2 examples of compounds that were found to be toxic in the clinic, and test them on human organoids. And the interesting point is that these were actually compounds where initially in the preclinical studies it was not discovered that they had toxic effect, but indeed with using human hepatocyte-derived organoids, we can indeed detect this already preclinically. Whereas actually the other 2 compounds also had some toxicity issues, but they were immune system dependent toxicity, which in this particular case, although I'll come back to it later, because we use a system that does not include the immune system, of course, we do not see any toxic effect when using them here. So basically, what this means is that we have these organoid systems that on the one hand, when we started out with discussing cancer, we can use viability tests to test drug responses in different tumors and in different patients. And on the other side, we have, because of these cells being human and being so close to patients, when we use the healthy cells, we can use them for toxicology studies, but also studies of inflammatory diseases like IBD or genetic disease. I didn't show that here other than the cystic fibrosis example, but so cells that are relatively healthy and normal genetically, and they can be grown in the differentiated cells, and the stem cells together will provide us a good picture of what the disease looks like. For the last bit of the presentation, I want to show another recent example. This is a picture from Anne Rios at the Prinses Maxima Center in the Netherlands, showing the co-culture system of T cells and organoids, a very important one at the moment, of course, because of the importance of the immune system in oncology and the new drugs that are actually treating this. So what we see here is in green an organoid with, in this case, targeting T cells that kill these organoids. The point of this already a couple of years old slide is that indeed it shows that we can co-culture these cells. And then here a more important picture is then when we use this was in specifically genetically manipulated T cells, where we have in these cells show that they target indeed specific tumor cells. On the top, we have a healthy organoid. And as you can see that hopefully in the right top corner, you see one organoid that is not targeted by these cells clearly. It's a healthy cell, so these CAR-Ts should not be targeting this. But now when we go to the bottom, in this repeating image, we see that there's the healthy one again, the intact one. But now the tumor of the same patient, and indeed it is specifically killed. So we see two things here. One, that we can co-culture organoids with immune system, the T cells, and show the killing of these organoids, of these tumor cells by the T cells. But also that we can show this difference between the healthy cells and tumor cells, and from the same patient, importantly. So in this case, we see that the healthy cell of that same person does not respond to the treatment as the tumor does. And you see that here in the summary again. But then for a larger group, so we actually have on the left here in imaging again, the top one patient, the tumor without and with treatment left and right, and then next to it normal organoids which are not responding to these T cells. And the bottom another patient, same thing. So the tumor is specifically killed, but the healthy cells are not. And then to the right, and that I think is an interesting point as well. So these are T cells that from which genetically it can be assumed that they should be able to target all of the tumor cells of this particular type. But what you see if we test a panel here, represented by about 10 different patients here with the different groups of black dots. So each small group of black dots is one patient. And what we see that on the left, we do see a group of patients that is not responding, and then we see an increasingly good response, measured in this case by interferon gamma secretion as an indirect measure of the response. So we see that there's an increasing response of the tumors to the T cell, or actually called the T cells to the tumor, and then with the open dots, the healthy comparison of the same patient not responding. So we do see this heterogeneity in responses, whereas although we wouldn't know the genetics behind it, we see a part of the patient not responding and another part that is responding. So I think this is the point, and that is also summarized here. So what we now have, now taking the organoid technology again, where we stand at the moment, we have shown that for many samples that we can generate these disease models that are genetically stable, that from a scientific perspective should indeed represent the patients. And indeed, when we do these comparison to the clinical studies, to the clinical results, we see a direct correlation between patients and tumor. Whereas at the same time, we can use we can use these samples in a group, in a biobank. So we can not only from a single patient, but in a group, we can have an idea of what happens to a treatment when we test it, we'll test it in a clinical trial. And so this picture indicates in a circle, as opposed to on the left a more traditional development from chemistry to cell lines to animal models to clinical trials, which leads to this failure to respond and cost inefficiency in the development of drugs. Because more in the circle, we can now connect the patients to the preclinical work. We basically have a system that allows us to test both clinically and preclinically the same type of patients, the same patient group, and it allows us to integrate the data, but also hopefully to much more efficiently develop new treatments. So thank you very much. That is what I wanted to show you today of the work that we've done at the HUB. This is here in this beautiful building here in the sun. Thank you very much, and